Good afternoon, everyone. Can I just check if you guys can hear me okay?
Yep.
Yes.
Yes.
Yeah.
All right. I'm just gonna see if anyone needs admitting into the session. Then I'm gonna share my screen. Then we can get started. Right. Can I just get you guys to confirm if you guys can see the slides?
Yep, all good.
Awesome. It's admitting into the session. All right, hopefully that should be fine. Okay, so the session today is talking about the introduction to neuroscience. So it's a very basic lecture today, just touching upon the background of neuroscience and I guess the surface level information that you guys would need to know going into this course. So my name is Fahad Ismail, and I've been a lecturer for neuroscience for about 8-9 years now. So if you guys have any questions, feel free to email me and I'll be happy to give you guys further support. Sorry, I'm just admitting everyone into the session so no one will be missing out. Great. Okay, so just to start off this lecture, I'm just going to give you guys a brief overview of neuroscience. So neuroscience is essentially defined as the study of the brain and the nervous system. So it actually encompasses all the different network that is associated with the brain and how it propagates to the rest of the body. What's really striking about the study of the brain and the nervous system is the fact that it can explain a lot of different aspects of our human nature, behavior, and function. So this actually involves explaining, our curiosity, expression of pain, pleasure, movement, reasoning, learning, memory, emotion, and even madness itself. So the origins of neuroscience is dated back to the into our prehistoric ancestors, where the brain has always been depicted as an organ that's vital to life. However, in the prehistoric era, the heart has been known to be the seat of the soul and also responsible for memory. Which is now we know that's not a true fact in terms of the role of the heart in itself. And what they've shown in the prehistoric times is that one of the surgeries that has been done in humans where they've actually drilled holes into people's skull. And the reason for this is that they've actually used that method called trepanation to actually alleviate certain brain maladies, including headache. And what they've shown by doing trepanation is that the skull is able to show signs of healing. So as we've evolved our understanding of neuroscience, one of the investigators that looked and studied neuroscience is called Franz Joseph Gall, who introduced the concept of phrenology. So basically phrenology is links the bumps of the surface of the skull and associate this with specific personality traits. So in this case, it's saying that specific structural changes, the structural features of the brain actually defines specific parts of our personality traits. In this case, if you have specific bumps in, let's say, the parietal area, if you look at the diagram over here, it tells you that you have more self-esteem compared to other individuals, okay? But as our understanding of neuroscience have actually evolved over time, these theory has actually been disproven, okay? So during the 19th century, there were four main major insights that we've had as our understanding of neuroscience have actually evolved. So the first one is that the nerve actually acts as wires. So what this means is that the nervous system has been determined to actually generate electricity, and the nerves that constitutes the framework of the nervous system actually conducts the electricity that is being generated by the brain and propagate this along the entire nervous system. The second bit is that the different parts of the brain perform specific functions, okay? So it's something that we will go through later on, where I'm gonna highlight specific areas of the brain and what they're associated with. The evolution of the nervous system. So we've kind of defined, the brain as the main organ, but you also have peripheral structures that are associated with the nervous system, including the spinal cord, and also the nerves that actually stretch out into the periphery of our body, okay? And lastly, one of the things that were defined in the 19th century is the identification of the basic functional unit within the nervous system, which is called the neuron. So now we're going to delve into the neurons and the neuroanatomy. All right. So there are two main cells that are associated with the nervous system. You've got the glial cells and also the neuronal cells. The glial cells are the cells that act as a support within the nervous system. So what they do is they actually provide insulation, support, and nourishment of neurons, okay? But the main player in the nervous system is actually the neurons itself, where they actually partake in information processing, sensing of environmental changes, communication changes to other neurons, which is done through synaptic transmission, and also command the body's response. So whether this is displaying specific types of behavior, these are actually mediated by the neurons themselves. Okay. If you delve into or hone into the structural components of the neurons itself, you could actually see that it is mainly composed of three different framework. In this case, you've got the microtubules, the neurofilament, and the microfilament as well. They have their own specific diameter, but all in all, these fragments actually constitutes the bone of the neuronal cells. Zooming out from the previous slide, You can see in this diagram, you've got a standard neuronal cell, okay, which is mainly divided into 3 sections. So you've got the dendrites, okay, you've got the axon itself, and you also have the axon terminal towards the end, okay. The axon in itself have different further components. So you've got the axon hillock, which is this area. So the axon hillock is actually the site wherein graded potentials actually sum up together to generate one big action potential that runs down the neuron itself, okay? You also have the axon proper, which is the middle part of the neuron, and also the axonal terminal towards the end. It is important to note that different sections of the neurons would have their own differences in terms of the components within the cell, okay? And this is mainly listed on this slide. So if you look at the axonal terminal, these actually don't have microtubules in their cytoplasm. They have the presence of synaptic vesicles, which are preloaded with neurotransmitter that is used to communicate to the postsynaptic cell. You also have the abundance of membrane proteins and also a large number of mitochondria, which is used when releasing the synaptic vesicles into the synapse. The dendrites themselves act as the antenna of the neurons, okay? So I guess if you look at the space in this area before the dendrites, you would have another neuron here, okay? And that neuron actually communicates to this neuron through the dendrites, okay? So the synapse is essentially the space in between 2 neurons, and that space is actually used to communicate as a means of communication between the 2 neurons through synaptic transmission. So synaptic transmission is composed of a transformation of signals from electrical, which is depicted by the action potential, to chemical, which is associated with the release of the neurotransmitter, and back to an electrical signal when the neurotransmitter elicits an action potential in the postsynaptic cell, okay? Synaptic transmission dysfunction has been associated with mental disorders. So one example that I could probably give in this situation is the onset of depression, okay? So it is really an important aspect of the nervous system. Going back to the glial cells, so depending on where the glial cells are located, you can actually find 2 types of these cells that supports the neurons. So you've got the oligodendroglia, which is mainly present within the central nervous system. And you also have the Schwann cells that are present within the peripheral nervous system. Their function is mainly to insulate axons by providing the myelin sheath, okay? So you've got, in this diagram, there's an axon that's covered with myelin sheath, which plays a role in trying to increase the transmission of signals along the axon themselves. Another thing that I wanted to point out here, if you have a look at this part of the diagram, you could see that in between the myelin sheath, there's actually a space called the nodes of Ranvier, okay? And this part of the axon is actually exposed and is composed of many ion channels that elicits or helps the propagation of signals along the axonal terminal. Moving on to the division of the nervous system, it is mainly divided into two sections. So you've got the central nervous system, which is composed of the brain and the spinal cord. And you also have the peripheral nervous system, okay, which is the neuronal connections outside of the brain and the spinal cord itself. If you focus on the peripheral nervous system, it is further subdivided into two parts. You've got the autonomic nervous system and somatic nervous system. The autonomic nervous system is the network associated with controlling our organs and glands, which is further subdivided into two more sections called the sympathetic division and the parasympathetic division. The sympathetic division is associated with our fight or flight response. So picture yourself being in a jungle and you encounter a lion. Your sympathetic division will activate, which will contribute to your fight and flight response by running away from a predator. Okay. The opposite of that is the parasympathetic nervous system, which is activated during our calm behaviors and also digestion. Moving on to the somatic nervous system. So this is where the network associated with voluntary movement, okay? So our skeletal muscles in particular is controlled by this network. It is mainly divided into two parts. You've got the sensory or afferent nervous system, and you also have the motor or efferent nervous system. All right, so this is a standard side view of what the brain looks like. And the main components here that I would like to highlight is the cerebrum, okay? You've got the brainstem and also the cerebellum. If you look at this brain diagram from underneath, one of the features that you can see is this bridge over here that connects the two hemispheres of the cerebrum, and this is referred to as the corpus callosum. If we look at the cerebrum itself, you can see in this diagram that it is mainly divided into several lobes. So you've got the frontal lobe, which has been shown to be important in mediating motor movements and also anticipation. We've got the parietal lobe, which has been implicated to be important in the somatosensory system. So whether it's our perception of touch and also the perception of pain, the information associated with those are actually processed in this area. The occipital lobe has been shown to be important in processing visual information. So all of the information that we perceive from our eyes actually project in this area of the brain, okay? And lastly, we've got the temporal lobe, which has been shown to be important in the processing of auditory stimuli and also processing memory as well. The next bit is talking about the meninges. So the meninges are layers in between the skull and the brain itself, which has been shown to be important in cushioning impact of the head to protect the brain itself. Okay, so the main meninges that we need to, I guess, remember for the purpose of this lecture are the dura mater, arachnoid mater, and also the pia mater. Okay, and pretty much this is the only thing that you would need to know from this slide is just being aware that there are specific layers surrounding the brain that collectively work to cushion the impact of the brain. Moving on to the next section, we have the ventricular system, which is shown in this diagram over here, okay? And the ventricular system is actually filled with a specific fluid called the cerebrospinal fluid. This cerebrospinal fluid is actually derived from the capillaries that actually project into the brain itself. And the composition of this fluid is actually controlled by a barrier called the blood-brain barrier. Okay, and one of the take-home message with this slide is looking at the flow of the cerebrospinal fluid along the ventricular system. Okay, and you can actually see how it circulates around the brain and also into the spinal cord itself. So the next bit is looking at the blood-brain barrier. So the blood-brain barrier is mainly defined as a highly selective permeability barrier that separates the circulating blood and the extracellular fluid in the central nervous system. It plays a major role in trying to regulate the molecules that actually go into the central nervous system. And it's a means of actually providing a protection that kind of restricts the harmful substance that can actually go into the brain itself. There have been specific cells that have been shown or demonstrated to be present in the blood-brain barrier, including endothelial cells, basal lamina, and also astrocytes. All right. So this slide over here is just showing you the different areas of the brain that have been defined over time. So I'm not going to spend too much time on here, but I'm just going to highlight a few areas like the motor region, okay, is mainly constituted by the primary motor cortex, or area 4. You also have area 6, which is the premotor cortex or the supplementary motor cortex. You've got the sensory regions like somatosensory cortex, which is present in the parietal areas. The visual cortex, which is present within the occipital area. The auditory cortex present within the temporal area, and the gustatory cortex, which is actually hidden. So you can't see it in the surface level, but if you open up this groove over here, you could actually see the gustatory cortex, okay? So moving on from the cortical areas, you can actually see the diencephalon, which is the posterior part of the forebrain. And the diencephalon is mainly divided into two sections. You've got the thalamus and the hypothalamus. The thalamus has been shown to be important in relaying sensory and motor signals from the periphery to the brain, or the other way around, and has been shown to be important in processing consciousness, alert, and sleep as well. Although the hypothalamus is confined in the same proximity as the thalamus itself, the function of the hypothalamus is very different from the thalamus itself. The hypothalamus has been shown to be important in linking the nervous and the endocrine system, okay, and also has been shown to be to play a role in homeostatic control, which includes the maintaining specific variables within optimal range, such as temperature, hunger, attachment, thirst, fatigue, sleep, and stress. Okay, so the best example that I can give you here is temperature. So whenever we expose ourselves in different environmental stressors, such as going out with no jacket in a cold, our body actually adjusts internally to be able to maintain our body temperature within the optimal range. And this is mainly controlled by the hypothalamus. All right, beyond the diencephalon, we'll see the mesencephalon. So the mesencephalon is also referred to as the midbrain, which is composed of the tectum, tegmentum, and the cerebral peduncles. So these structures have been associated with the diffuse modulatory system in our brain, and this is something that we will delve more into the course as we go along. Okay, but for now, given that we're only talking about the introduction, that's the only thing that you guys would need to know for now. Beyond the mesencephalon, we have the rhombencephalon, okay, which is the lower part of the brainstem. So the first two structures that you can see here is the medulla oblongata and also the pons. So the medulla oblongata has been associated with autonomic function. So it controls heart rate, respiration, coughing, swallowing, vomiting, and sneezing. And any issues or any lesions within this area can actually disrupt this critical function of our body. Okay. If you look at the pons has been shown to relay information between the cerebrum and the cerebellum. Okay. And any disruption in the pons can actually affect its function in relaying these information between the cerebrum and the cerebellum itself. Looking at its function, it has been associated with processing information associated with hearing, equilibrium, taste, facial sensation, and general motor control. The next bit associated with the rhombencephalon is the cerebellum, which has been shown to be important in motor control, mainly in coordination of movement, precision, and timing. Okay, so if you actually hit somebody from the back of the head, it can actually affect their movement coordination. Okay, so that's where the cerebellum actually comes into play when it comes to contributing to our motor movement. Okay. Other evidence associated with cerebellum has shown that this is also important in attention and language, as well as fear and pleasure responses as well. Okay. So the next bit is talking about the limbic system, which is an important part of the brain function and has been associated with processing our general behavior, our display of emotion, motivation, memory, and also other functions. One of the things that you guys would need to know about the limbic system is that it's actually a system of different structures that work together in order to regulate these functions. Okay, so the main structures that are involved within the limbic system are the cingulate gyrus, components of the temporal lobe. You've got the hippocampus as well. Okay. So as you go down from the brain to the spinal cord, you could actually see that the spinal cord is mainly divided into different areas. So you've got the dorsal area, the lateral area, and also the ventral area of the spinal cord. Okay. The role of the spinal cord is essentially acts as a conduit between the brain and the periphery or the rest of our body. Okay, so it actually links the brain with the periphery, including the skin, joints, and our muscles. The dorsal area is mainly associated with carrying sensory information from the periphery to the brain. And the ventral area has been demonstrated to carry motor information from the brain to the periphery. The next bit is talking about the cranial nerves. So we have 12 cranial nerves that we have in the brain. And the cranial nerve is actually a nerve that connects the periphery to the brain without going through the spinal cord itself. Okay, so these 12 neuronal network includes the olfactory nerve, which is important in carrying smell information. Okay, we've got the optic nerve, which carries sensory information from the eyes to the brain. You've got the oculomotor nerve, which carries information from the brain to the eye that controls eye movements. The trochlear nerve is also associated with eye motor movement as well. You've got the trigeminal nerve, which carries somatosensory information from the skin to the brain. Abducens nerve innervates the lateral rectus muscles of the eye. You've got the facial nerve, which controls the facial muscles in our face, okay, and is responsible for us executing different facial expression. The eighth cranial nerve is the auditory and vestibular nerve. As the name suggests, this is associated with carrying auditory information from the ears to the brain, but also carries vestibular information, which is associated with our balance in a given space. Okay. The ninth cranial nerve is the glossopharyngeal nerve, which is a nerve that's mixed of both sensory and motor information that goes into and outside of the brain. The 10th cranial nerve is the vagus nerve, which carries parasympathetic information to the organs. Okay, so a good example would be the heart. So the vagus nerve actually plays a role in controlling our heart rate. The 11th nerve is called the accessory nerve, which innervates the sternocleidomastoid and the trapezius muscles that we have in our neck that stretch down in the back. And lastly, you have the hypoglossal nerve. So this is composed of both intrinsic and extrinsic innervation that innervates the muscle of the tongue. All right, the next bit is talking about interesting aspects of what is called the connectome. So the connectome is actually generated by a device called the MRI, where they've actually implemented a procedure called the tractography. So what the tractography actually does is it actually generates a comprehensive map of the neural connection. And the main focus here is to actually generate a detailed wiring diagram of the brain itself. So from what you can see in the slide is what the wiring will look like if you try to project it in the brain itself. And the things that you can take away from here would depend upon the different scales that you will be applying when viewing this radiography. So you could look at it at a micro scale, which shows you the neural connection between two neurons. You can look at it in the mesoscale, which shows you the connection of different neuronal population. And if you look at it at a macro scale, you can actually see the neural connections within systems and organs itself. In terms of the application of the connectome, it is important to note that it's not only confined to the brain itself, you could also project this along the periphery as well, and how it innervates the different organs that we have in our body. All right, so the next bit is talking about the membrane potential. So just to give you guys a brief introduction in membrane potential. So all cells in the body have a negative resting membrane potential that ranges from -5 millivolts to -100 millivolts, depending on the cell itself. Cells can either be defined as electrically excitable, such as neurons and muscle cells, but there are also cells that are non-electrically excitable. A good example here would be skin cells or squamous cells. The membrane potential is defined as the voltage across the neuronal membrane at any moment, and it is mainly abbreviated as Vm, as you can see in the slide. And the resting membrane potential that we have for a given neuron is actually -65 millivolts, okay? And what this means is that if you look at the inside of the cell, you could actually detect that the charge of the cell inside compared to the outside is more, it is actually more negative with respect to the outside of the cell. So the resting membrane potential maintenance hinges on 3 different factors. So you've got the concentration gradient of ions across the plasma membrane. This is most importantly seen with sodium ions and also potassium ions. It is mainly defined that there's a high level of sodium ions outside of the cell and high levels of potassium ions inside the cell. But the resulting difference here is that the inside of the cell is a lot more negative as opposed to the outside of the cell. Another factor that allows for the maintenance of the resting membrane potential is the presence of ion channels and ion pumps within the plasma membrane. So you can actually see these as the purple structures in the slide, okay? And you also have certain resting ion permeability through leak ion channels. Okay, so these are ion channels that are open at any given time, which allows for the movement of both potassium and sodium ions across the membrane. So this slide just shows you the concept of talking about the inside of the cell being more negative as opposed to the outside of the cell. It is important to note that when we're talking about that concept, we're not really talking about the charge of the cell inside of the cell. It is mainly associated with the differences between the space just in between the membrane, okay? So if you look here, so this is the inside of the cell and this is the outside of the cell. When we're talking about the inside of the cell being more negative as opposed to the outside of the cell, we're only talking about this segment, okay? And also this segment of the outside of the cell. We're not really talking about the entire cytosol itself, okay? And the transfer of ions in between these two sides is only associated with this space over here. All right, so in terms of maintaining the resting membrane potential, so there are different type of transport that actually controls this. So you have the simple diffusion and facilitated diffusion. Both are actually referred to as passive transport, mainly because they don't actually use energy when activating this receptor proteins, okay? The active transport part, which is shown in this part of the diagram, actually utilizes ATP, which gives this receptor energy to open and close, okay? And this is referred to as the active transport. So the ion channels is a receptor structure, a protein structure embedded within the plasma membrane that allows for ions to passively cross the plasma down their concentration gradient. So there are different aspects here when talking about the property of a given ion channel. So you've got the conductance, which is defined as the ease with which ions can diffuse through a channel. And you also have the concept of gating kinetics. So when it comes to gating kinetics, this is mainly attributed to three different types. So you've got the voltage gating, which is controlled by electrical potential. Okay, you've got the chemical gating, which is controlled by the binding of a specific molecule or chemical into the receptor protein. And you also have the mechanical gating, which relies on the deformation of the plasma membrane that allows for the opening and closing of an ion channel. Okay. The concept of selectivity is basically associated with the channel's ability to allow specific ions to diffuse within its protein structure, okay? So this slide, it just shows you the different type of gating mechanisms that each ion channels use. So we've got the ligand-gated ion channel, which relies on the binding of a specific chemical. In this slide, you can see the neurotransmitter is bound to this receptor that allows for the opening of this ion channel, okay? So in this case, when the neurotransmitter binds to the receptor, it allows for this structure to open up, and the sodium ions can cross the plasma membrane. Mechanically gated ones are those that respond to mechanical pressure. So in this case, when you have a pressure being inflicted in this area of the plasma membrane, it allows the ion channels that is close in proximity to actually open up, which allows for a molecule to actually cross the plasma membrane. And lastly, we've got the voltage-gated ion channel, which mainly responds to the onset of an action potential in its periphery. And when you have the arrival of an action potential in this membrane, this part actually gets dislodged, which allows for a molecule. In this case, you have the sodium ion channel to go through the ion channel. And this part over here talks about specific ion channels that are always open, which is referred to as the leak ion channels. And this actually allows for the continued transfer of potassium and sodium ions across the plasma membrane. Okay, so these are always open. Further to that, there are specific receptor proteins that are located in the plasma membrane that works as an active transport. Okay, so the role of having an active transport is really there to compensate for the sodium and the potassium leak channels. So when you have these ion channels that facilitates the ongoing transfer of sodium and potassium ions across the membrane, there has to be a mechanism that actually readjusts and maintains the resting membrane potential at its optimal level, which is the negative 65 millivolts that we've highlighted before. The role of the sodium and potassium ion pump is to maneuver 3 sodium ions out of the neuron and also getting 2 potassium ions into the neuron, which is depicted in this diagram over here. Okay. So the resulting effect here is that there is a net loss of positive charge within the cell, which makes the inside of the cell more negative as opposed to the outside of the cell. So this slide just shows you a way to actually model the resting membrane potential in an in vitro system. Okay, so in this case, you can actually model it by having 2 chambers that is separated by a modeled plasma membrane over here. And one of the things that you would notice is that there would be a diffusion across the plasma membrane. In this case, you have the movement of the potassium ions from one chamber to the next. Okay, and the movement that you can see here is actually associated with reaching equilibrium to make sure that everything will actually stay in balance when it comes to what's on one side of the chamber as opposed to the other side of the chamber. What's important to note here is that one of the defining factors that defines how ions actually move between 2 chambers is the presence of specific ion channels. So in this case, when you have a potassium ion channel that's depicted in this part of the diagram, you could actually see the movement of the potassium ions, which actually leads to a more positive charge on this chamber as opposed to the other chamber. Okay. All right. So the equilibrium potential is the potential with which the movement of the ions needs to go through in order to reach an equal level of charge in between the 2 chambers. Okay. It is mainly defined by the Nernst equation, which is shown here. Okay. But for the purpose of this course, we don't really need you guys to actually calculate the equilibrium potential. This is just a way to introduce this concept to the students. So when neurons actually communicate with one another, the first step in initiating this communication is through signal initiation, okay, which involves the generation of an electrical current in the form of a change in the cell membrane potential brought about by the alteration of the membrane permeability to the ions, and also the alteration of various ion concentrations across the membrane. So we spoke about the idea that the inside of the cell is more negative as opposed to the outside of the cell. But if there's any changes associated with this, for example, the potential of the cell becomes more positive as opposed to being more negative, then this can actually lead to a generation of a potential, okay? And when that potential is actually generated, that is referred to as the generation of the graded potential. So a graded potential occurs when a gated channel is opened or closed, altering the ion flow through a membrane. Changes in the ion and charge distribution causes voltage changes to the resting membrane potential. So in this case, it's making it more positive, as you can see in this diagram over here, okay? And when you reach a specific threshold, this graded potential can actually turn into an action potential itself, okay? It is important to note that the graded potential doesn't only reflect a change of the membrane potential from being more negative to being more positive. There's also hyperpolarizing graded potential that changes the membrane potential from, I guess, for example, negative 70 millivolts to, let's say, negative 80 millivolts, okay? The only difference between this effect and this effect is the fact that this graded potential will cause the cell to generate an action potential, while this graded potential actually prevents action potentials being generated. All right, so in terms of the polarization, two terms that you guys would need to know is the concept of depolarization and hyperpolarization. So depolarization is a reduction of the membrane potential. So it's actually making the cell more positive as opposed to having the inside of the cell being more negative as opposed to the outside of the cell. Hyperpolarization is when the inside of the cell becomes more negative compared to the resting membrane potential. The generation of an action potential is mainly defined by the principle of the all or nothing principle, which is mainly highlighted in this slide over here. Okay, so there are four factors that constitutes the all or nothing principle. The first one is that the depolarization must reach a threshold value to generate an action potential. Okay. The second one is that the action potential magnitude remains the same along the length of the axon. The third one is that the movement of the action potential triggers the next section of the axon to fire. And lastly, it is just a concept that various stimuli can induce a change in the potential of the neuron that leads to the generation of an action potential. So there are 3 points that you guys would need to remember when it comes to the all or nothing principle. It is important to note that if the stimulus is too low and it doesn't reach a specific threshold, it actually doesn't lead to an action potential, okay? The other thing that needs to be remembered when it comes to the all or nothing principle is the fact that if you reach that specific threshold that allows for a generation of an action potential, the magnitude of the action potential will always be the same size, okay? And lastly, when it comes to looking at the magnitude of the stimuli itself that leads to the generation of an action potential, instead of the magnitude of this deflection over here, it is actually defined by the frequency of the action potential that is being generated within that neuron, okay? The next bit is looking at the structure of the action potential itself. So this is a standard depiction of what an action potential looks like, okay? Having taught this for several years, one of the things that I've noted is that students don't really, I guess, take on board what an action potential looks like. I've seen them try to draw action potentials in the form of an ECG wave with the P wave, QRS complex, and also the T wave at the end. So I'm just gonna highlight this to you guys that it is important to be aware of what an action potential looks like, okay? Which is depicted in this slide over here. So there are a few sections that you guys would need to know when trying to demonstrate what an action potential looks like. So you've got the onset of the stimulus over here, which changes the membrane potential and makes it more positive, okay? Once it reaches a specific threshold, this actually leads to an overshoot of the potential of about 30 millivolts, okay? And once it reaches the peak, it actually goes down again, okay? There's an overshoot down here that makes it more negative than the resting membrane potential, but the leak channels allow for it to adjust back to its optimal resting membrane potential, okay? And this last slide here is just talking about the different ways that the axon actually relays information within the nervous system. So one of the things that I've highlighted before is that there are axons that are insulated by myelin sheaths, but there are also neurons that actually don't have the myelin sheath. And there are two ways that action potentials are being propagated along these cells. So the first one is what they call as the continuous conduction. And this mainly occurs in the transmission of action potentials along unmyelinated axons. But you also have the saltatory conduction, which is the propagation of an action potential within axons that has the myelin sheaths on it. So in this case, you have the skipping of an action potential, okay, from one node to the next. And what's important here is that the saltatory conduction is more quicker as opposed to the continuous conduction, which relays information a lot slower within the nervous system. And that's pretty much it when it comes to the introduction of neuroscience. I've just left my details here. Should you require any further clarification on the topic today, so feel free to reach out and I'm happy to have this discussion offline. We have about 1 minute left in our session, but if you guys have any questions now, feel free to ask. Otherwise, I'm happy to close the call.
Someone actually posted a question on the chat.
Yeah, I'm just looking at the chat now. All right, let's see what we can do here. Okay, is it mandatory? The answer, I believe, is no. This session is recorded and will be made available to the students. So if attending the live session is not very helpful to your learning, I would say that just playing the recording is adequate enough. Next question, do we have to know each cranial nerve carried to the brain? It is important to know the specific function of the cranial nerve. Some of them are pretty straightforward. For example, the olfactory nerve, it mainly carries smell information that you detect from your nose to the brain, where the brain actually processes that information. The follow-up question in regards to attending lectures, whether it's mandatory. I'm not sure about the tutorial. I would say the tutorial is shown to have more, is more mandated compared to the lectures, but that needs to be discussed with your tutors, okay? Anyone know if there's a chat on Facebook? I'll let the students answer that question. All right, everyone. I don't think we have any other questions.
Actually, I had one question. It's regarding the
I don't think I can hear you loudly.
Oh, it's okay. I kind of just solved my question myself. It's all right. Thank you.
Okay, great. Awesome.
Hi.